Hi, this is chapter 2, and we didn't really have any sort of formal lecture. I wanted to go over a couple of things in this chapter. Almost all of the slides that were originally posted are in this, but I just want to highlight some of the key words that should resonate with you so that you understand what they mean and can use them, and then there's some problems at the back. All right, so this is really the chemistry. And you guys can read the PowerPoint, so I'm not going to go through slide by slide. This just lays out what is going to be important to study in the chapter. One of the most important things about this chapter is it points out features of the carbon atom. So carbon plays a central role in all of life science and biology. It has this valence of 4, so it can form 4 chemical bonds with other atoms. And so it's a very flexible atom, and it's present in the biological macromolecules, nucleic acids, proteins, carbohydrates, and lipids. And to reiterate what a covalent bond is, this shows just different valences. Like I said, you can all go through these. Looking at energy, the amount of energy needed to break these bonds. This is a nice little graph showing the strength of hydrogen bonds is way down here, covalent bonds here, double and triple, of course, higher energy within that bond. All right, so the term hydrocarbons is something I want you to be familiar with. Let's see if I can get my pointer. Hydrocarbon is just a chain composed of carbon and hydrogen. And so what that should bring to mind are things like hydrocarbon chains in lipids, or the fatty acid chains in fat molecules, for example. Those are composed of just hydrogen and carbon. They are important for fuel, and they are important structurally for membranes. Now, they are not soluble in water, so this is one of the big sticking points of hydrocarbon chains, and that's what composes lipids. So lipids are not soluble because of the large component of hydrocarbons. All right, some different other molecules. Take a look at the atoms that are important in biological compounds. Carbon, hydrogen, atom, nitrogen, and occasionally you'll see phosphorus. You see the phosphate group in phospholipids that form the membrane. You see phosphate groups in nucleic acids, for example. Sulfur. You'll see sulfur occasionally in proteins for sure, because it's an atom in the amino acid cysteine, and that's what forms disulfide bonds. These individual atoms also form what's called functional groups. So these are little groups that give molecule personality. For example, the hydroxyl group, the OH group in the amino acids serine and threonine, that really affects the properties of those amino acids. And these are some important groups. Okay, polarity. So we'll talk a little bit about water, but think about this term electronegativity, and how a particular atom in a compound can be more electronegative or electropositive. Another term, polar bonds. What does that mean? Well, it means that the electrons are not shared equally between 2 atoms. Think about the water molecule. You have a very electronegative oxygen and partial positive charges on the atoms. So those are polar bonds, not equally shared. And I still haven't been able to figure out how to get those. Know what an isomer is. These are stereoisomers where they're superimposable configurations. It's just a term to add to your science vocabulary. Whoa. An asymmetric carbon. So each of the 4 bonds that those electrons on the outer valence, they associate with different compounds. All right, there's a good bit here on water. I mentioned polarity of water, and with water, it really allows water to have some unusual properties. Cohesiveness of the water molecules that interact to form liquid water or ice or whatever. It has a temperature stabilizing capacity or heat index, and it has major ability to bring other compounds into solution. So that's called a solvent property. All right. Again, with the water, the polarity. Oops. I don't know how to make that go away. I guess it'll just go away. I've talked enough about those. All right, so I'm posing a question to you. How many hydrogen bonds can one molecule of water form? Okay, this oxygen, hydrogen, hydrogen, these 3 atoms form the molecule water. Now, molecules of water can associate in the liquid solution we think about as water. And it does this through all of these hydrogen bonds. So here is, well, let's take this one here. This water molecule here interacts with how many other different molecules? So you can see the oxygen interacts with 2 hydrogens, whereas each hydrogen interacts with other oxygens. So 4 hydrogen bonds per water molecule is what is typically seen in solutions. All right, some more features of water. All right, so I combined a couple slides here to show you this cool property of water, and this is related to the surface tension. And this transpiration process causes water to be taken up by cells in the roots, and water will ascend into the upper levels of the tree and then be evaporated from the surface of the cell. And all that is due to the surface tension of water that allows these water molecules to be cohesive. Okay, so these 2 terms, hydrophilic, hydrophobic, I'm assuming you know what those mean at this point. All right, so I want to talk a little bit about spheres of hydration. So this is what happens when you have ions, cations or anions in solution. They're very soluble, and they're very soluble because they are polar. This has a positive charge, and it attracts the partial negative charge from neighboring water molecules. And so that's what keeps that sodium in solution. Chloride, it's the opposite. The chloride ion is negatively charged, and it is kept in these sort of cages by the partial positive charge of the hydrogens in water. The importance of selectively permeable barriers. You guys know that some things can get in, some things can not get in, at least as far as diffusion. Okay, what is it? A membrane is a hydrophobic permeability barrier, and it really defines what a cell is. It defines the outside and the inside of a cell. And the membrane is composed of phospholipids. So that's what you made in class with the 3-dimensional structures. Glycolipids, which is like a membrane lipid, only with a carbohydrate attached. And membranes typically also contain proteins that can go through the membrane. They also contain hydrophobic molecules, cholesterol, which is a steroid, and this is what we see in animals. In fungi and plant, there are some other molecules, but cholesterol is an important component of biological membranes. the lipids are amphipathic and that they have hydrophobic and hydrophilic regions. Here's an example of the phospholipid that you would have made in class. Now, this is all that was really shown in the little model you used. You made 2 fatty acids. This is the core glycerol molecule that's used to make phospholipids. Here's the phosphate, but there's also usually a charged group on the other side of the phosphate molecule. So this polar head group is not just phosphate, but it's linked through phosphate. All right, so what things can get in, get out? Well, these charged ions cannot get in, cannot diffuse in. Glucose and sucrose, large uncharged polar molecules can really sometimes come in if they're uncharged. Small uncharged can more easily. You can see this diagram has most of the glucose, sucrose staying out of the cell and some coming in, whereas this one, ethanol and water, some of it does not go in, some of it can diffuse out. And oxygen and CO2 are typically able to diffuse through. They're small nonpolar molecules. Okay, so these ions and charged proteins or charged molecules and large proteins and things like that, they are able to get into a cell, but they are not able to diffuse across the membrane. So something more targeted has to be present in the membrane to allow it, to allow those components to cross the membrane. Ions must be transported across the membrane by specialized transport proteins. And we'll certainly learn about that later on, but know that, yes, they can get across the membrane, just they can't diffuse across there. And they are able to come through the cell membrane through these particular specific channels, or sometimes carriers are involved. So those are pretty complex structures. We've already talked about some about macromolecules. Now, proteins, nucleic acids, polysaccharides are typically described with more similarity. Lipids have some features that are not in the same category of those, but they're nonetheless called a macromolecule category, but they're synthesized somewhat differently. And here's an example of small building blocks of organic molecules that can form these structures: cellulose, protein, DNA. You know, when you see a figure like this, it's really helpful for you to take a minute and orient yourself. So you've got arrows that are coming down. You've got a cell here. You've got small organic molecules up here. So what it's showing you is how these are organized into macromolecules, and then sometimes supramolecular structures. And sometimes these are composed or are contained in organelles, and together those can form the essence of a cell. So this is a table you could draw yourself easily. The inorganic precursors, what of these are contained in the organic molecules that form the building blocks of these macromolecules. So be sure you know those. That's really helpful to understand more complex parts of cell biology, if you can get that now. Different kinds of macropolymers. And they're making a distinction between macromolecules that are informational versus have a coding function. And I am going to leave this for you to read. It's nothing tricky here. All right, so one thing I wanted to talk to you about is the monomers that form the polymers. Now, typically the monomers can't just be a glucose or amino acid or something like that to form a macromolecule. Typically, they have to be activated before they can associate to form a macromolecule. Typically, the energy to couple the monomer to a carrier molecule or activation molecule is provided by ATP. And I already mentioned the polarity of macromolecules. Here they're describing it directionality, so the ends of polymers are different. Here's some examples, and you can see that these are dehydration reactions where water comes out, but initially the monomer is activated on so that it can associate with another monomer, and additional steps can be added as the polymerization process goes through. So there's different kinds of carrier molecules. This is not necessarily an activation. For protein synthesis, amino acids, so this is in the cell, amino acids are linked to tRNAs. And I'm sure you remember how the tRNAs bring the amino acid into the ribosome, and the amino acids are joined there. Well, the amino acids that come into the ribosome are activated, so they are attached to this tRNA that allows them to then be polymerized. And we talked about dehydration of polymers. So a water molecule is removed as a polymer forms, but to break those down, you have a hydrolysis of that event. So to hydrolyze something, you have to have a water molecule added to the mix. Now, some structures and some proteins or macromolecules can be assembled through their own information. So they can just form seemingly spontaneously, just the information in that macromolecule. Others, and this applies specifically to proteins, they need a little help to then fold into the right structure. And there are a lot of classes of proteins structures called chaperones. And these protect the protein as it goes through all these different funny little steps, trying to find the perfect, the lowest free energy state of that protein to assume. So using molecular chaperones is kind of the opposite of self-assembling of a structure. All right, different kinds of non-covalent interactions. Okay, so where do you find nonpolar groups? And let's stick with the protein because we've talked about proteins a bit. Where would you find a valine or a leucine or isoleucine? They are hydrophobic proteins, and they really don't like to have to deal with water. So typically, as a protein is being synthesized, you've got a grouping of hydrophobic amino acids. They're going to interact with each other, but it's not really like a bond, but they like to huddle, and so that they don't have to deal with water. So hydrophobic amino acids tend to be in the interior of a cell, and hydrophilic amino acids tend to be on the outside of a cell. Proteins fold in the cell. Polypeptides fold into proteins, but a protein can also be natured, and that means they lose their biological activity. But sometimes they can renature. This is an example. Here's a native molecule all folded up, presumably functioning properly. And here's a denatured molecule. So maybe it's been heated, loses its structure. In certain conditions, you can change the buffer it's in and it can renature. I discussed a little bit of molecular chaperones. So self-assembly can happen with a macromolecule. It can also happen with certain structures, ribosomes in particular. All right, so there are some questions here. Which of the following statements is true of biologically important chemical bonds and transitions? Does it take about half as much energy to break a non-covalent bond as it does to break a covalent bond? The energy to break a double bond could be plotted on this graph between an NC bond and the phosphocreatine. It takes less energy to break one of the phosphate bonds in ATP than it does to break one of the carbon-hydrogen bonds in methane. Well, since breaking of a phosphate bond in ATP is really critical to cellular function because ATP activates so many things, I would say that it would be the latter. Suppose you're studying a new and unusual form of life that contained a fourth type of macromolecule, which is a long polymer. What would probably be true of this chemical? Well, they're not saying it's a protein, so it's not necessarily composed of amino acids. We don't know that it's going to be a fat soluble, and we have no information about what it functions, if it functions as a pigment. What about this? Produced by dehydration synthesis. So we looked at carbohydrates, nucleic acids, proteins, all of those polymers formed by dehydration, removal of water to associate the two monomers. Probably the right answer there. So if you're breaking down a macromolecule, we just looked at dehydration. If you wanted to break apart two monomers essentially in a polymer, you'd probably need to add that water back in to get those apart. So you would add an oxygen and two hydrogens to the ends of those associated monomers. What cellular macromolecule is most certain to include phosphate? Think about that. Do proteins typically have phosphate? Does DNA have phosphate? Carbohydrates, lipids? What do you think? So specificity is a big umbrella of biology. And there's a lot of basic concepts, and one of them is specificity in general. So receptor proteins are very specific for their ligand. Enzymes are very specific for their ligand. So what's the difference? Each of them interacts with a ligand, and that causes some sort of conformational change. A receptor protein would lead to a signaling event, an enzyme might lead to a catalysis event. So what are they differing by? Receptor protein is encoded by DNA. What would an enzyme too? So you're looking for differences. Receptor protein has directionality. Wouldn't an enzyme too? They're both proteins. Enzyme can have quaternary structure. Well, there's no reason why a receptor protein wouldn't have quaternary structure too. Certain receptors. An enzyme might catalyze hydrolysis of a specific, this is a very strong bond, see, oxygen bond. Well, I never heard a receptor that does that. Receptors are more involved in signaling, whereas enzymes are involved in catalysis. So this catalysis word is your clue there. What does multimeric mean? Consists of a single subunit, more than one subunit. Protein and RNA can copy itself. Multimeric. Okay, barley cells synthesize maltose, whereas mammary cells synthesize lactose. Therefore, barley cells contain genes that directly specify the linkage of alpha-D-glucose to alpha-D-glucose in maltose. Mammary cells contain genes that directly specify the alternation of galactose and glucose in lactose. Barley cells contain a gene that itself encodes an enzyme that catalyzes the synthesis of maltose. Mammary cells contain a gene that encodes an RNA molecule that catalyzes the synthesis of lactose. Well, they're telling you that both of these polymers, maltose and lactose, are enzymes. So it's going to be an enzyme that specifies the linkage. I think this is the last one. So you have to answer this question, you have to know what maltose and lactose is. And I'm not going to tell you that, but you can figure that out. Okay, DNA is a polymer of blank. So this is a short answer question. You'll have short answer questions in your exams. These subunits are blank to each other through a phosphodiester bond, double-stranded RNA, and double-stranded DNA, sorry. They are also blank to nucleotides on the opposite strand. This is a great question. It has to do with the macromolecule nucleic acid. So DNA is a polymer of what? Nucleotides, bases, or nucleotides? So what did you make in class? Well, some of you made those. Nucleotides. And you put nucleotides together, the sugar of one nucleotide to the phosphate of another to make a chain. Not a helix, but a chain. And that bond, when you put the sugar onto a phosphate group and the next nucleotide, that bond is called a phosphodiester bond. Okay. The subunits are what to each other through a phosphodiester bond? Take a look at that. You think that's a non-covalent bond that is between those? Remember, it's going to be through a shared pair of electrons. All right, so then you've got all of these nitrogenous bases on one side, and to make a double helix, they're going to interact with another strand, and they associate with the nucleotides on the opposite strand. So are those bonds going to be hydrogen bonds? Well, apparently so, because that's your only option here. Now, what's the advantage of this hydrogen bond over, say, a covalent bond? Well, a covalent bond is really strong and really pretty permanent. Hydrogen bonds can be interacted with to break those. So if you're going to replicate DNA, you've got to unwind your helix. You've got to open up that DNA strand. All right, why are carbohydrates more hydrophilic than lipids? Okay, remember we talked about lipids have these long hydrocarbon chains, these long fatty acids. Whereas carbohydrates, many of you remember seeing the hydroxyl groups on the carbohydrates, and you were calling the hydroxyl group a sugar or an alcohol. Well, these OHs are common to carbohydrates, and these OHs are very soluble. This oxygen can form lots of hydrogen bonds with water, so it's certainly more solvent. Okay, yay. So that's it for a quick run through for chapter 2. And I need to get this posted, and thank you very much. You have an exam on Wednesday, and we'll be able to go over some of this between now